Hi, thanks for watching my talk. So I'm excited to tell you today about some work that I've been doing with Terry Dial and David Reznick on local adaptation in skull shape of Trinidadian guppies in response to differed trophic ecologies. So although the guppy is one of the most widespread freshwater fishes in the world, it's really gained fame for its natural history on the island of Trinidad, just off the coast of Venezuela. And so what's really interesting about the geography of Trinidad is that it's split at the top. There's a mountain range running across it. And so what this mountain range does is separate out different river systems in which the guppies are genetically distinct and kind of create isolated replicates of a natural experiment. So here I've highlighted the Yarra River and the Aripo River. And what's really interesting is that in each of these rivers, there are natural barriers, such as waterfalls, that although they allow guppies to get past them, they prevent most other fish species from getting above them. So that means if we look at sites above and below these barriers, we find completely differing ecologies. So one of the most famous ways in which people have studied this is looking at predation levels. So below the barriers, there are high levels of predation on the guppies, and above the barriers, those predators aren't able to get there, so there's very low predation on the guppies. And there's been a lot of work done on how this affects the life history traits of the guppies and the mating coloration of the males. But there's also been some work done on how this affects the rest of the ecosystem around them. So here we see kind of gut content analysis from the guppies, both in high predation environments and low predation environments. So the y-axis tells you what portion of the food that they're eating comes from a certain prey type. So on the left, the high predation fish are primarily eating invertebrates, and this means mostly free swimming things. So it's mostly a limnetic feeding style. Whereas on the right, in low predation environments, they're eating mostly detritus and algae. And these are food types that will be on the bottom, so they require more of a scraping or a picking kind of motion. So if we look back, we can summarize this to say that low predation environments create a benthic feeding environment, and high predation environments create a limnetic feeding environment. So if we think back to the different sampling sites that we had thought about before, we see that this creates two replicates of a system in which there is an ancestral limnetic population that has then moved up into a benthic portion of a river and now has a chance to respond to that change in ecology. And what I think is really interesting about this is that this benthic limnetic divergence has actually been kind of a famous axis of evolution in fish before. So when we think about other benthic limnetic divergences, the classic example is cichlids from the Great Lakes in Africa. So here we have two different species from Lake Tanganyika. The top one is a limnetic feeder, and the bottom one is a benthic feeder. And what we see is that in the limnetic species, they have a very elongate skull, and that they have a very shallow craniofacial profile, whereas the benthic species has a more compressed skull with a steeper craniofacial profile. And what's really interesting is that both within and between lakes, we see a huge amount of convergence to these morphotypes within each feeding space. So we were really interested to see if guppies would go through a similar process as they're exposed to a novel benthic environment. So this brings us to our main questions. So the first question is simply whether the guppies have modified their morphology in any way in response to this novel benthic environment. Then if we do see a difference in their morphology, we're curious if the differences are mostly genetically based or to what degree they are plastic. And finally, we're really interested to compare any results we find here with the cichlids and see if the guppies are changing their skulls in similar ways to the cichlids in response to the same sort of feeding divergence. So to do this project, the first thing we did was collect wild guppies in Trinidad, and we collected them in these 4 environments that I mentioned before from 4 different populations. We preserved half of them immediately to represent the wild morphology. Then we took the other half back to the lab and raised them in a common garden environment so that we could examine the genetic basis of morphology several generations down the line. With all these preserved fish, we micro-CT scanned them and then placed a set of 3D landmarks in order to complete a geometric morphometric analysis. So here I'll show you the landmarks that we used. You'll notice that we only placed landmarks on the left half of the skull, and this was to remove any effects of asymmetry within the skull. You also know that we haven't outlined all of the bones of the skull, and this is because at this stage in this project, we're largely interested in kind of the overall gross morphology of the skull, and we haven't gotten yet to looking at individual elements. So once we have those landmarks, we can do our geometric morphometric analysis. So this begins with a generalized Procrustes analysis, and this is to remove the effect of position, rotation, and size, such that we're only left with shape and can compare the shape of different individuals. Then we do an allometric correction, and this is just because there tends to be some differences in size between the different populations that we looked at. So we want to remove the effect of size or developmental age from our data before we compare benthic versus limnetic. And then finally, we analyze group differences using both linear modeling as well as principal component analysis to allow us to visualize the differences. So when we start to look at our data, we will mostly use principal component analysis plots. So here the x-axis is the first principal component axis, which explains the most variation that we can find across all of our fish. And the y-axis is the second principal component axis that explains the second most amount of variation in a way that is linearly independent from the first. So the way to read this plot is that 2 points that are close together represent two individuals that look very similar, and 2 points that are far apart represent individuals that look very dissimilar. So what we can do is we can put on first, we have the wild-caught limnetic fish, then I've added the lab-reared limnetic fish. Now we can add the wild-caught benthic fish, and finally the lab-reared benthic fish. But we're really interested in the possible shapes that they can take on. So what I've done is outline the area that each of these groups covers. And so here in blue, you see the possible shapes that limnetic fish can take on. And in red, the possible shapes that benthic fish can take on. And then if we put this into a linear model, we find that there actually is a statistically significant difference between benthic and limnetic fish when examining both the wild-caught and the lab-reared fish. So there does seem to be some level of local adaptation going on, but we still don't know whether that's genetic or plastic. So next what we do is look at our lab-raised females. And again, every portion of the difference here should be based on the genetics of the two different populations, and there shouldn't be an environmental effect. So if I show you similarly outlined areas, we see that the benthic and the limnetic fish do separate out. And so it does look like there is a genetic component to this benthic-limnetic divergence within the guppies. And then that leaves us with just our wild-caught fish. So the wild-caught fish, their morphology is going to be a result of both the genetics underlying their development as well as plasticity in response to the environment that they were raised in. So if we expect some differences because we know there are genetic differences, but if we see that they're even more diverged from each other than the lab-raised fish were, it indicates that there's also a plastic effect. So when we look at the area included, the area covered of this morphospace by both the benthic and limnetic fish, we see that again, they are kind of disparate and that they are statistically separate from one another. So we really can separate out benthic and limnetic fish in the wild. What I think is also interesting, and I won't go into it too much, but I thought it was worth mentioning, that we also plotted out the two different river systems, and there seems to be a significant difference between those as well. So we're not sure if this has to do with these fish evolving with a different genetic background, or if there are more differences between the environments than we've given it credit for, but just kind of an interesting thing to note. So the last thing we want to do, now that we know that there are differences, we want to know what those differences are. So I'll show you again quickly our landmarks so you can kind of orient yourself to the skull. And then what I'll do is compare the average landmark position of the benthic fish with the average landmark position of the limnetic fish. And I've magnified the difference between them by 5 times to help illustrate that. What I've also done is highlight in gray the regions between homologous points. So how you can read this is the bigger the areas in gray, the bigger the differences are between the benthic and limnetic fish. So some specific results that I'll point out are that on the benthic fish, the frontal bone on top of the head is more dorsal, The maxilla at the front of the head is more posterior, and the preopercle has moved a bit ventrally. So we can think about this again with our overall guppy skull and start to compare it to the cichlids. So if you remember, we had this divergence between limnetic and benthic cichlids. So here we'll focus on the benthic cichlids because what we've looked at in the guppies is the changes that happen as they move into the benthic environment. So if we focus on these first two arrows and think about raising the frontal bone and moving the maxilla posteriorly, this will have the effect of creating a steeper craniofacial profile, just like we saw in the cichlids. And if we focus again just on the maxilla, and as that moves posteriorly, that's compressing the skull, again like we've seen in the cichlids. So it looks like there is some real convergence going on, at least at kind of a broad morphological level. So just to summarize what I've shown you today, the first question was whether guppies show any difference between benthic and limnetic environments. And the brief answer is yes. We were able to differentiate between benthic and limnetic fish when looking at all of our fish. The next question that we had was to what degree these differences were genetic. And although the differences are less clear, there still is a significant effect of the niche. And so there is a genetic component to differentiating benthic and limnetic fish. And then we asked whether there was also a plastic effect. And it looks like this is also the case because when we looked at the wild-caught fish, they were further diverged from one another than the lab-reared fish. And finally, we asked whether the guppies were convergent with cichlids and how they responded to the benthic limnetic divergence. And in terms of the gross morphology, it looks like the answer is yes. But we're really excited to continue this project because we feel like now we've established the fact that there is a benthic limnetic axis of divergence in guppies. And so now we'd like to know whether the function of the benthic guppies is converging with the function of the cichlids, whether the specific morphological elements that change to create these gross morphological changes are convergent, and even whether the developmental mechanisms that they use to cause this change, whether there's convergence there. So with that, I just want to say thank you for watching the talk, and if you have any questions, please feel free to contact us or to just leave them in the chat box. Thank you.